Thanks a lot. The work I'm going to tell you about today is it's going to be a quick summary of, or the culmination of 5 or 6 years worth of work that I've done primarily as a graduate student at the University of Washington. And right at the front, I want to acknowledge 3 of my key collaborators on different parts of this work. They are Daniel Schindler at the University of Washington, Mike Adams at the USGS in Corvallis, and Craig Williamson, who's at the University of Miami, Ohio. And what I want to tell you about today is really, it's an evolution of my thinking about how we go about evaluating different kinds of anthropogenic stressors for species of conservation concern. And clearly we're all concerned about amphibians here today. And I think that the nature of having this sort of applied question for species of conservation concern really brings our uncertainty about conclusions that we make about any one factor or suite of factors into sharp focus, so that ultimately what we want to do is make predictions or conclusions and explicitly acknowledge our level of uncertainty or certainty in each step in that process, and critically evaluate where and when we think different stressors are important. Okay, so just to start more generally, even more generally than that, which is that I think that one of the greatest challenges to applied ecology lies in understanding how particular environmental stressors or anthropogenic disturbances, or combinations of them, affect species that by definition occur across incredibly heterogeneous environments. Okay, so how do we capture that incredible heterogeneity when by tradition and out of experimental and observational convenience, we tend to study these things at relatively small spatial and temporal scales? So how do we go from studying them at relatively small spatial and temporal scales, but still wanting to make predictions at these broad emergent levels that ultimately things like conservation and management are happening at? So landscapes, regional species dynamics, assemblages of species dynamics across these huge swaths of land that are nearly impossible to track experimentally, and that are far beyond the ability of any one researcher. Okay, so what I want to tell you about today is this process that I went through in trying to evaluate the importance of anthropogenic ultraviolet radiation exposure of a suite of alpine amphibians for the Pacific Northwest, but really use that as a case study for just thinking through how we might actually go about making these conclusions and being very careful about the level of our certainty at each step. Okay, so I don't have to tell this audience that anthropogenically increasing ultraviolet radiation due to stratospheric ozone depletion has received a tremendous amount of attention in the context of global amphibian declines. And when I started graduate school about 6 years ago, it was the leading hypothesis for declines in the Pacific Northwest, especially for the suite of subalpine and alpine amphibians that occur in protected natural areas, which is primarily where I worked, which is up and down the Cascades and in the Olympics of Washington. There have been over 100, to date, there have been over 100 individual studies done evaluating the relative sensitivity of almost as many species of amphibians. And what we know from those studies is that some amphibians are incredibly acutely sensitive to even ambient levels of ultraviolet radiation, where you get big mortality. You can measure big mortality in the face of just ambient levels of UV. And then there are other species that are relatively insensitive even to elevated levels of ultraviolet radiation. So we've invested a lot of time into doing these sorts of studies. Wait for it. I just put this talk together last night, so I'm not actually sure what slide comes next, but it's not the same one. Can I get some help with the next slide? Okay. There we go. Okay, so just to illustrate this, these are made up data, by the way. Here's a landscape in Olympic National Park in Washington. And imagine if you were a researcher, if I were a researcher, and I went out to my favorite site, and I wanted to do an exposure experiment to figure out whether or not ambient levels of UV were influencing the populations that I was interested in. I might go out to this site and shield a number of embryos. In this case, I'm going to tell you a story primarily about embryos, but this could be true for larvae, other aquatic life history stages. You might shield a number of embryos or egg masses from ambient levels of UV and then assay something about the survival, the performance, the growth of those individuals through time, and on a relative basis compare whatever that metric is to see whether or not shielding those embryos or larvae from UV had a measurable and significant impact. And if it did, if these were the data, so that minus UV you get a big bump up in survival, we might say, okay, well UV is probably exerting a pretty strong influence on the individuals in this site. Okay, but what we really want to know, again, striving for this generality, is what are the particular suite of conditions that occurred in this one site that led to UV being important here? Okay, and even more broadly, what does doing the experiment here tell us about the importance of UV for individuals occurring here or at all of these other sites, or all of the sites that I couldn't possibly put in one picture? And really what we want to do is ask the question in a different direction, which is, what are the conditions that lead to UV having strong impacts on individual species? And then, how frequently do those conditions occur across broader landscapes? Okay, so the story I'm going to tell you really deals with this suite of subalpine and alpine amphibians that are predominantly pool breeders in the Pacific Northwest. And I don't have time to go into the details for all these, but I've tried to pay very close attention to what's going on for almost all of these species. Today I'll tell you primarily about cascades, frogs, and long-toed salamanders. So you all know what the life history strategy is here, which is that they tend to lay their embryos in the water, and then they hatch out into some sort of aquatic larva, metamorphose into something sometimes in the terrestrial world. But our conventional wisdom says that embryos are probably in what we consider the most vulnerable environment, both because they're in this open, exposed place, and they have no ability to change their environment. Okay? Whereas larvae may integrate the signal of their environment over a different sort of spatial and temporal scale, embryos are stuck where they are. And so one positive thing that means is that the accounting of the UV exposures of embryos is a little bit more straightforward. Okay, so if we wanted to predict the exposure of lots of embryos across the landscape to UV, one of the critical things that we need to account for is something that limnologists have appreciated for upwards of 15 or 20 years, which is that the critical factor that's important for predicting UV exposure in any body of water is actually the concentration of aromatic dissolved organic matter, which dissolved organic matter is the byproduct of terrestrial plant decomposition. It's aromatic ring structures that end up as in a dissolved form in the water column, and different concentrations of DOM have a striking effect on UV wavelengths in particular and how they're transmitted with depth through the water, such that if you have a high concentration of dissolved organic matter in a single site, let's say, as UV hits the surface of the water, the way in which that UV is transmitted with depth is such that it's rapidly attenuated, so that maybe at 10 centimeters depth, these are actual data, maybe at 10 centimeters depth, nearly 90% of what hit the surface of the water is gone. Okay? And in a lower concentration condition, maybe at 10 centimeters depth, 80% of what hit the surface is still present. Okay, so what I want you to appreciate here is that small differences in the concentration of dissolved organic matter result in really big differences in the UV environment of aquatic things. So if we're interested in predicting the UV environment in the water, we need to know what the concentrations of these critical features of the water are. And we can do this, we can go out, this is just to show you that this is just a simple rate coefficient that we can estimate very quickly with collected filtered water samples. And what that means, what these relationships mean, are that we can go out to a large number of sites, collect water samples, and rapidly characterize the relative difference in the exposure environment of the water itself, giving us sort of the framework or the backbone upon which we then will evaluate sort of the generality of what we think UV impacts might be. Okay, so this is a slide that summarizes, like, I don't know, 5 or 6 years worth of work, which is to say that we went out to hundreds, literally hundreds of different sites in subalpine and alpine areas, protected areas of the Northwest, in Oregon, Cascades, and all over the Olympic Peninsula in Olympic National Park, As we go out and collect these water samples and go to all these different sites, we monitor those sites for the presence of breeding. We characterize the UV environment by collecting those water samples and lots of other things about those sites as well. And then in a subset of sites, we do things like map the specific embryonic distributions within sites. So we know where these species breed across these sites, and then within a subset of them, we know very specifically what the depth distribution and the local cover environment of all the embryos in these sites are actually like. And this is going to be really important in terms of accounting for all these other factors that are important in predicting total UV exposure on an embryo by embryo basis. Okay, so this is the slide that also summarizes 5 or 6 years worth of work, which is that in my evolution of studying this, I went from thinking about the lab-based physiological assays of the differences between these species to doing these big field-type surveys to try and map out the distributions and the particulars of their embryonic distributions. And the physiology tells us that these two salamander species are far more sensitive to levels of UV than are either of these two frog species. And then that's largely a function of their photorepairability. Some experiments I did with Craig Williamson that have just come out in the Proceedings of the Royal Society show this, if you're interested in more details. And following up on that, it turns out that these two sort of most physiologically sensitive species, if you look at the places where they tend to lay their eggs in the sites where they choose to breed, we found an incredibly interesting suite of patterns for these two species, which is that northwestern salamanders, as a function of the clarity of the water to UV alone, nothing else about the site, there is a very strong relationship such that as the transmission properties of the water go up, so it's more and more harsh of a UV environment, the depth at which they lay their eggs goes down. So they increase the depth at which they lay their eggs, presumably protecting them in some way from that increase in UV exposure, and that long-toed salamanders appear to modify the habitat in which they lay their eggs. So a higher proportion lay their eggs underneath impervious, light impervious surfaces as the transmission properties of the water go up. We're still working on teasing out the mechanisms behind those, but, and then we have all this field distribution data. Okay, so what I really want to say about this is just sort of background, is that we can take this sort of static information about basic physiological differences, their behavior and their distributions, and combine that with what we know are sort of the temporal dynamics of UV exposure in any one breeding season, and estimate the actual total dose of UV experienced by all these eggs in a large population. Okay, so the temporal dynamics of UV exposure are what you already intuitively know, which is that summer solstice is the highest flux of radiation from the sun, and on either side of summer solstice it drops off. So depending on when the window of breeding occurs for each of these different sites, you are exposed to a different sort of atmospheric level of UV exposure. That's important to account for. And also, additionally, is that each one of these sites, these are a pond by pond basis, just to illustrate this, is that their transparency through time to UV also fluctuates as a function of snowpack melting and temperature and precipitation and the availability of the pools of dissolved organic matter pulsing into these systems. So now if we know the window of when embryos of each of these different species are present in each of these bodies of water, specifically the bounds around that temporal window, then we can estimate how much UV reaches a particular depth in any one of those sites across that entire period. If we also happen to know the oviposition distribution across depths of all the embryos in each site, then we can, on an egg-by-egg basis, come up with an estimated cumulative UVB exposure per egg. Okay. So now, once we have those UVB exposures across a large number of sites, we can put them all together and say, on average, what are the conditions that most embryos of these different species with different underlying physiological sensitivities exposed to. So here are those distributions for what I was actually a lot of work to get for 22 sites for each of these two species. And these are frequency distributions of the total UV dose experienced on an egg-by-egg basis, tallied up across all those 22 sites. So you can see that they're strongly skewed for each species, with lots of them exposed to very, very low levels of UV, in part because if they're laid underneath something impervious, or they're laid deep in the water in a site that has high levels of dissolved organic matter, they're not exposed to much cumulative UV. Okay, but what do these tell us about risk? They don't tell us anything about risk, is the answer. Because what we need is to go on top of this where those sort of critical levels or critical zones, if you will, of sensitivity actually fall on these distributions. And then that's going to tell us something a lot more about the frequency at which we would expect UV to regulate individuals. Oops. Okay, so we go back out to the field. Here's a nice picture of the Olympic Peninsula. If you squint, you can see the boundaries of Olympic National Park based on the clear cutting that goes on outside of it. Okay. And so what I did is actually a series of just these simple egg incubation experiments where you hold eggs 10 centimeters below the surface of the pond, use simple screens to shield out some proportion of the UV, and then compare survival. Did this for where we had sort of 6 replicates per pond, 5 ponds per species, and a couple of species. And the ponds were chosen intentionally along a gradient of UV transmission properties of the water to give us that gradient in UV exposure through time. This is what it looks like. Very simple, nice little experiment. And then now this is, for each species, these are the sites where we did a full factorial experiment in each site. Okay, and now these are the effect sizes in each of those factorial experiments, where this is the mean difference in % survival between ambient and shielded embryos. So that if you have a big positive number, it means that shielding long-toed salamanders in this site resulted in a 40% increase in survival of those embryos. The stars denote statistical significance. You can see that the magnitude of the effect for these two species is very different, reflecting their underlying physiological differences. But that also the threshold or the zone at which UV, this is total dose of UV estimated that they experienced across that experiment, is also much lower for long-toed salamanders than it is for Cascades frogs. Okay, if we wanted to draw the sort of thresholds on here, one possibility is, I might need your help again. One possibility is to draw the line at the site that had the lowest level of UV exposure that was statistically significant. So it has a statistically significant effect on survival. So we would draw a line here, we would draw a line here. Yeah. Can you help me advance that slide? Probably means I should have compressed the pictures in this presentation. Thanks. Okay, so if we wanted to be sort of liberal, we would draw the line here for each of these species. But if we wanted to account for, again, our uncertainty in the fact that we don't think necessarily the threshold response is the same for every individual, and that necessarily this is exactly that level of UV that's associated with mortality and nothing below that is important, then we could draw the line at the highest level of UV exposure that was not statistically significant. So somewhere in here is a zone of sort of uncertain but potentially harmful UV, and we know that the red zone is harmful. Okay, now if we plot those onto these distributions, here's what we find. For Cascade frogs, 0% of all the, whatever, hundreds of thousands of eggs that we measured occur in that critical red zone, and less than 3% occur in that uncertain zone. For long-toed salamanders, the picture is pretty similar, where we have less than 1% occurring in this red zone of known lethality, and less than 10% in this sort of uncertain zone. It's important to remind you that the way I've done this is fairly conservative in the sense that I'm saying that anywhere that was statistically significant is this zone of lethality. The statistical significance doesn't necessarily equate to biological significance. These are the associated expectations for mortality, where we saw a 7% on average reduction in Cascade frog embryonic mortality and a 42% bump up for long-toed salamanders. So these are actually fairly conservative. So let me just say that one way that I think that we can sort of tackle evaluating different kinds of stressors and disturbances to species with very different ways of interacting with these heterogeneous landscapes is to take sort of this hierarchical approach of going across scales of biological organization and recognizing that if we do the experiment at only one scale and we make a conclusion at broader scales based on that experiment, that it's likely to be very context dependent. And what I think we're striving for as applied ecologists is to actually get better at our predictions and to force ourselves to ask the questions of whether or not what we contend is true and where and when we think it's true. And that's a hard thing to do, I think, in a world that sort of operates on timescales equivalent to like a graduate degree or an NSF grant, or publishing a paper, and everybody gets pushed to say things more generally in their papers than maybe our data warrant. So actually stepping back and critically evaluating this, I think, is critical for our progress as applied ecologists. I didn't get to talk about future projections, but let me just highlight this to say that it's likely that DOM concentrations are going to be more sensitive to future environmental change than our projections for UV distributions in the future, and that if we're interested in understanding UV in the future, is probably the key thing to appreciate as these distributions change. So thanks. Sorry for going over.